the answer to this question is it takes a lot of people to bring new drug combinations into clinical trials. Different researchers, different healthcare professionals, and ultimately big groups of people across Europe to run the trials themselves. And I'm not going to talk about that at all. I'm going to talk about some very specific strategies that we've been taking in the labs here in Leeds to try and identify new drug combinations that could potentially be evaluated in clinical trials. But some good news, just to keep remembering the good news. More children survive cancer now than ever before. So if we look at these figures back in sort of 1970s, there would be just 10% of children who survive their cancer. But now over 80% are surviving their cancer. So that is good news, and I think it's good evidence that research works. But these children that are surviving have got a number of health problems. We've just heard about some of the health problems with some of the treatments that they receive. So we need to think about how we might be able to overcome some of these problems for long-term health of patients. And obviously we want to cure all patients. I'd rather not have to do my job. I'd like all children to survive cancer and have an active life. So I guess the strategy for everybody in this room and anybody doing research, whether that's translational research in the labs or whether that's clinical research running clinical trials or looking at side effects and mental health, we want to save more lives and we want to improve the quality of life of patients. And so what we're looking for in the labs then is to find more effective treatments and kinder treatments, so we don't have the long-term toxicities that we see with the current chemotherapy. This big step change in outcome that you see here was really due to the introduction of chemotherapy like doxorubicin and Christine to, as Steve said earlier on, shrink these tumours, but often you don't always kill all the tumour, and so this is why sometimes patients have relapse, and then this can lead to progression. So I think Steve also mentioned this sort of pipeline to the clinic, and Jess talked about phase 1 trial. So I'm not going to talk at all about this part of the pipeline to the clinic, because that's not primarily what we do in the labs in Leeds. We focus on this preclinical part, and I'll talk about that a little bit more. But I wanted to put this slide up just to give you an indication of how expensive it is to get a new drug into care, but also how long it takes. So 10 to 20 years to develop a new drug and get a new drug is standard of care for a patient. £800 million. And if we look at the sort of rate, if I think about drugs that I might be investigating in the lab, that the 10,000 drugs, maybe only one of those will end up being used to treat patients. And the biggest problem is this preclinical stage. So the things that we're doing in the labs to identify drugs that might be effective in patients. So that's kind of where we're really focusing on what I'll talk a bit about today. There's a complex preclinical pipeline that's been established for many years using cell lines, which are cultures of tumour cells that have been in culture for 30 or 40 years, and then looking at drugs in mouse models. Some of these are useful strategies, but I think actually, to study the cancers, we need to be studying cancers from the patients rather than cell lines that have been cultured in the lab for maybe 30 years and will have changed and adapted because they've been grown on plastic dishes, basically, in an artificial environment. And that's really what we've been doing in Leeds. In my lab, we've been doing this particularly focusing on Ewing Sarcoma, which is the second most common bone tumour in young people. But the strategy that I'm going to describe could actually be applied to, in my opinion, any solid cancer. But what we've been doing in Leeds for 4 or 5 years now is receiving tumour samples from patients, from surgery, at diagnosis and sometimes at relapse. It's usually difficult to get tumour samples at relapse. But from Birmingham, Newcastle and Leeds, taking these tumours from the surgical centres, which are transferred to us in the post. They can be in the post for 3 or 4 days, and we can still manage to grow tumour cells from these samples. It's because these tumour cells are pretty aggressive and they're quite difficult to kill. So what we've been doing in Leeds, particularly led by a really talented postdoc in my lab called Ewing Sarcoma, is isolating tumour cells and also the microenvironment cells. So obviously within a tumour there are the tumour cells, but there are also supporting cells that help the tumour to grow. So we've been isolating these cells and propagating them in the lab and studying them within a few months of them coming out of the patient, not 30 or 40 years. We've been particularly interested in trying to develop drugs to kill the cells that cause progression and relapse, so cells that are resistant to treatment, the current treatments, and are also able to divide and self-renew, so are responsible for both localized and metastatic relapse. I'm not going to go into lots of detail to go into this slide, but what we do in Leeds is then, having established the cultures, we use multi-complex imaging systems to look at the tumour cells both in isolation, just on the plastic dish, which is kind of what you're looking at here. These are some tumour cells that are stained here with a cytoplasmic stain. This is actually a stain called DAPI that picks up the nucleus of a cell, so we can use this to count the cell number and then study the actual cell phenotype. We can look at lots of different markers and investigate what's driving these tumours. But we can also put these tumours into 3D structures, so into clumps, if you like, both alone but also with the microenvironment cells. So we're generating a 3D model of the tumour and using that information combined with information about the patient course, so whether the patient's had treatment, whether they've done well, and also the genomics and the transcriptomics, so the information of the actual genes and RNAs and proteins that are driving the behaviour of the cell. We can combine that to really understand at a patient level what these tumours look like. And Steve mentioned this idea of putting patients together in groups dependent on particular biomarkers. So a biomarker is basically something that will define a characteristic in a patient. So every time you get on the scales, you find out whether you've put on weight. So that's a biomarker of your weight. And we use biomarkers to stratify patients into different risk groups. So patients who are going to do well compared to patients who are going to do less well, with the idea that we might give patients who do well less treatment to minimise their side effects, but maybe more importantly, those patients who are, did I say that? Did I say less well? Did I say the other way around? So the patients who do less well, we give more treatment, and the patients who do well, we give less treatment to minimise their side effects. So this is one of the things that we use these cells for, and we've identified some specific markers, some specific markers based on hypotheses looking at the actual genes that are mutated and abnormal in these cultures in patients, and then finding drugs to target those particular genes and pathways. And this is just showing some examples. These two drugs are actually inhibiting an enzyme called aurora kinase A, which affects LIC, which is a protein that causes proliferation and is highly expressed in Ewing's. So these are potentially interesting candidates of drugs that might be used to improve outcome for Ewing's patients. So this is one way in which you use the cultures, but most of our efforts over the last 4 years have been focused on trying to find drugs that will kill these cells that are drug resistant and self-renewing, so the cells that are responsible for the local and metastatic relapse. And just sort of try and take you through the hypothesis of the work here. that's been funded by Little Princess Trust and CCLG. As we've already heard, tumours often respond to treatment. Solid tumours will respond to chemotherapy, they'll shrink. And so you might kill all these sort of orangey yellow cells, the cells that are responsive to chemotherapy with the initial treatment. But you don't kill these cells that are resistant to the treatment and self-renew. And so the tumours regrow. They can regrow with the same genetic material, the same signature, or they can sometimes change. The treatments can induce changes in the cells, but effectively you've seen a response in the patient, but you haven't cured the patient. So our idea is that we can find a drug that will kill these grey cells, the cells that cause progression and relapse, and combine that with chemotherapy and radiotherapy potentially, we'd be able to kill all the tumour and cure more patients. To actually find drugs that will kill these grey cells that are causing progression and relapse, we've taken a drug repurposing and repositioning strategy. So this is really taking existing drugs that are used in other cancer types or in other diseases even, and looking to see if they'll kill those grey cells. There's lots of advantages to doing this because already these drugs have been in patients. We know lots about the safety of them. We know actually how they work usually, not always, but usually. And so we can think about different drugs that might be combined to target different pathways that are driving aggressive behaviour of tumours. And they're usually licensed for use. So this can be speeding up the process of getting a new drug into a clinical trial. So our hypothesis really is if we can find a drug that kills those grey cells that's caused progression and relapse and combine it with chemotherapy, we might be able to reduce the amount of chemotherapy we give and so reduce the toxic effects that we see, the long-term side effects that we see with some of the chemotherapies, but also improve the cure rates and outcomes. So this is just an example of, I haven't given the details, but each of these little marks is a different drug or an inhibitor that's been put onto a whole panel of these little grey cells that are driving relapse and progression. And some of these are inhibitors of targets, and some of these are actually driving the actual target to have a therapeutic effect. And this is just an example. You can see all these drugs here below this line are more effective than doxorubicin at killing actually the total cell population, but also at killing these little grey cells. So these are attractive potentially for evaluating in patients. And one of our top hits that we've looked at is a drug called napibucacin. It's widely available, it's very cheap to buy, lots of information about safety. And on this graph, there's just a plot of the number of cells of this axis and then increasing concentrations of this drug. In each of these lines is a different grey cell population, so a different population of these cells causing relapse and progression from different patients. And what you can see here is actually we can kill these grey cells with this drug called napibucacin. And if we combine this napibucacin with chemotherapy, so in this example here, similar idea, number of cells against concentration of drug. If we look at napibucacin here, the blue line, if we combine that with ifosfamide, which is a drug used in treatment of Ewing's, you can see that you get this synergistic effect. So both drugs together have a better effect than one drug alone. And so by using napibucacin, and this is also true with other chemotherapies, doxorubicin and crystalline things, we might be able to use napibucacin in combination with these chemotherapies to reduce the amount of chemotherapy we give, therefore the side effects, but still have the efficacy. And some of these drugs, for some of the cells that we've investigated, actually the napamecasine has an effect. This is viable cell number concentration. Napamecasine has an effect where doxorubicin or some of the other drugs that are used in treatment routinely don't have effect. So potentially this is a drug that could be effective in patients that don't respond to chemotherapy. So this is a sort of strategy that we've taken in Leeds to try and identify drugs that we can accelerate into early phase clinical trials, see if they might improve outcomes for some patients. And I think that actually using these patient-derived primary cultures can provide a number of opportunities to improve outcomes, identify novel therapeutic targets, these biomarkers of risk, dividing patients into different groups for different specific treatments, drug repurposing, which I'm talking about, but they can also tell us some things about the biological insight. But for me, the biggest challenge of this approach is how many models do we need to be able to find a drug or a drug combination that's potentially going to be beneficial for all Ewing's patients? Because Ewing's, as any cancer, is highly heterogeneous, and we all know that patients have different outcomes even though they've got the same diagnosis. So what we're currently doing is trying to get funding to allow us to characterise more of the models that we've established from patient tumours, and look at the models we've developed in the lab and compare them to the tumours in the patients and see how representative our models are, both of the individual patient, but then taking a broader look across a range of patients and models, see how many models we might really need to study to get a very convincing combination that might be most effective, so that we make that 10,000 molecules go into one drug more like, well, 1-to-1 would be great, but reducing that attrition rate. And we're looking to actually work with Genomics England to get sequencing data that's been generated through NHS England strategies. So I hope I've given you a bit of a feel for the kind of strategies we're taking in Leeds to try and identify new drugs that might go into clinical trials. We're extremely grateful to patients for giving us samples. If we didn't have these samples, we couldn't do this research. And obviously to the surgeons who collected the samples and sent them to us, but also to all the funders and the other collaborators that we work with that trying to move some of these drugs forward.